So last time we introduced the general knowledge on virology, and we learned the viral structure and also how they cause disease, how they make new viral particles in host cells, that's in our cell. And this week, we'll talk about some typical viral pathogens, uh, including both Tuesday and Thursday. We'll talk about the important viral pathogens. We'll start with influenza viruses. You know, we are experiencing the pandemic, and coronavirus attracted our attention a lot. But in the history, coronavirus uh, didn't cause a pandemic as big as influenza virus. Later, we'll introduce the history of influenza pandemic, which is much worse than the current pandemic we are experiencing. That happened about 100 years ago, from 1918 to 1919. That pandemic is much worse than the pandemic we are having now. So influenza virus, that's what we will learn today. Yeah, we are experiencing the pandemic. We talk about coronavirus a lot. We talk about COVID-19 a lot. But also keep in mind now that for now, at this moment, it's also a flu season. So from October every year to next January or February, this is a flu season. So always every year there is a peak of flu infection. That's infection by influenza virus. And and it seems that we are kind of confused. We are not paying proper attention to flu. We think that a flu is just a severe cold and everyone can recover. But that's not true. Sometimes flu infection can be fatal. It can kill a lot of people. To prevent flu or to beat the flu, we need to do a lot of things to keep everyone healthy. And in these different measures, including washing hands, avoid touching eyes and nose and mouth, and staying home if you are sick, and also get the flu vaccine shot. Of all the measures, the flu vaccine is very important. But the problem is that all over for the whole population, only a very small portion of the people would like to get a vaccine, because those vaccines are not covered by the vaccination plan. You have to pay for it, and not everyone get a vaccine every year. Later, we'll talk about the flu vaccine. Okay, influenza virus. There are several types, 3 types influenza viruses identified, including influenza types A, B, and C. And of those, type A and type B are the most frequent pathogen for human. They can cause disease with symptoms similar to common cold, but much worse than common cold, such as febrile, so that's high fever. That fever is usually higher than a common cold. And also respiratory illness. They cause infection in the respiratory tract. And also with systemic symptoms all over the body, including like muscle pain and also a lot of other symptoms. Maybe they are also also cause complications. This is the structure of influenza virus. Well, basically they look like a sphere. So they show us like spherical, roughly spherical structure. That's because influenza virus is enveloped virus. There is a membrane envelope on the outside of the virus. So that causes the virus look like a ball, roughly a ball. The size of the sphere is about 100 nanometer. So that's the diameter of the virus, about 100 nanometer. Of the three types of viruses, influenza A and B virus contain single-stranded negative sense RNA genomes. It contains 8 separate segments. Like in this figure, you can see it contains 1, 2, 3, 4, 7, 8. 8 RNA segments. And altogether, the 8 RNA segments make one genome of the virus. So this is type A and type B. While influenza C virus contains 7 segments of RNA. And C virus doesn't express an enzyme called neuraminidase. And neuraminidase, short for NA, is important for infection in our body. This is the structure of the virus. Okay, I can see students are still coming in. Okay, because the virus contains RNA segments, those RNA molecules are separate from each other, like shown in this figure. This A virus contains 8 segments, labeled in dark, while virus B contains also 8 segments, labeled in white. What will happen if both viruses infect the same host cell? Co-infection, that's what we meant. Infection by both A and B at the same time. What will happen? That means that the A genome and B genome will mix together in the host cell. When new viral particles is formed, the new viral particle might contain both A and B. There will be a mix together, and this mix together we call genetic reassortment. For example, the new viral particles, like this one, it contains fragments from both A and B. The black ones are from A, while the white ones are from B. So this we call the genetic reassortment. But some virus might be the similar one from the beginning, like this one. This is a B, this is a A. But a lot of viral particles contain RNA fragments from both A and B. This phenomenon, we call it genetic reassortment. And because of this genetic reassortment, the new viral particle might get new properties, new creatures. The new viral particle might carry properties or characteristics from both A and B. And there is a mixture of genome, and there might be new antigens expressed by the new viral particles. And this makes the influenza virus very difficult to be immunized because the virus is changing so fast. Every year, the virus is evolving. It's different from last year. It's different from the year before. So every year, we have to make a new vaccine. We have to estimate, we have to predict, we have to guess what will be popular this coming year and make a vaccine targeting on a new strain. So this is genetic reassortment. And now we are talking about the genetic, the viral structure and composition. This picture showed us the schematic drawing of influenza virus. In the middle, it contains RNA fragments, and they are bind to proteins. And on the outside, there is a membrane layer, that's the envelope. And on the envelope, there are some spikes that's similar to the spike protein of coronavirus, but those are different proteins, including HA and NA, and also smaller proteins and M protein and NS2 protein. Those are different proteins. And the two most important viral proteins on the envelope are HA and NA. HA is short for agglutinate, is short for hemagglutinin. So that's short for HA, hemagglutinin. And NA is short for neuraminidase. HA can agglutinate red blood cells. And with this property, we can use red blood cells to detect the existence of influenza virus. But keep in mind, not only the influenza virus can agglutinate red blood cells, some other virus can also agglutinate red blood cells. HA can help the virus to bind to the susceptible cells and can stimulate the production of neutralizing antibody. So this is HA. Well, NA, neuraminidase, that's one enzyme, the sialidase enzyme, which can remove the sialic acid from glycoconjugates. So it is important to help the new virus to leave the host cell surface. So after infection, new viral particle will form, and the new viral particle will leave the host cell through budding, because they are enveloped virus. So they leave the host cell through budding. And the final step for the new particle to leave the host cell requires the neuraminidase, because it can catalyze the sialic acid group so that it can break it down and leave the new viral particle. This HA and NA, the two important envelope protein. Here's a little bit about the pathogenesis of influenza virus. The transmission is usually through airborne droplets. So when a person carrying the virus cough or sneeze, they can release the airborne droplets, which contain the viral particle, or through direct contact. The contaminated surface will contain the influenza virus, and if someone touches the surface and touch their mouth or nose or eyes, and that will also transmit the virus and infect the person. The incubation time is about 1 to 4 days. So that's relatively variable and pretty short. At the very beginning, the virus attacks mainly the upper respiratory tract, like nose or a little bit deeper. So that's the primary infection site or site of entry. Viral infection can cause damage of the epithelial cells in the respiratory tract. And this can lower its resistance to bacterial infection. So usually or sometimes influenza virus infection can cause complication of viral infection or bacterial infection. That's because the virus damaged the epithelial layer, and that epithelial layer is more susceptible to bacterial infection. Okay, including staphylococcus and streptococcus and also others. Severity, usually a flu is severe, and it's much more dangerous than a common cold, and it's dangerous especially for the very young and the elderly. The very young, for example, the infant, the child, or the elderly. Like think about your grandparents. When they get old, their immune system is not working well and is weak than before. They might be more susceptible to influenza virus infection. And also those people who are immunocompromised, if they have trouble with their immune system cannot work well, that's a problem. And also for people with heart or lung disease, influenza virus might be dangerous. Symptoms, and we don't have to spend a long time here, including like fever, headache, myalgia, cough, rhinitis, or even vision problems. So that's a lot of symptoms. Some of them overlap with common cold, but usually the symptoms are much worse than common cold. Complications. It doesn't always happen, but sometimes during a flu infection there might be complications, including like pneumonia. That's due to some infection in the lung. And those complications may be fatal, particularly in elderly persons with underlying chronic disease. And as we mentioned that influenza virus can lead to secondary bacterial infection, including staphylococcus infection, streptococcus infection, and also the Haemophilus influenzae infection. The last one is a bacteria. Okay, so this is the complications by influenza virus infection. During the influenza infection, the immune system wants to handle it. So your immune system wants to handle it, but that not always, your immune system doesn't always work well. That's because influenza virus is an RNA virus, similar to coronavirus. Coronavirus is also RNA virus. And one property of RNA virus is that their genome is not stable. Mutations can happen with a faster speed. So they can get point mutations much easier than DNA virus. And those mutations, at the beginning, it happens only for some points. That mutation we call the minor mutation or minor antigenic change. Mutation in the genome can change the gene sequence and also can change the protein sequence. If it happens only like a smaller scale, it can cause minor antigenic changes in like HA or NA. This is due to the accumulation of point mutations in the gene, resulting in amino acid changes in the protein. But this change is a minor change, not like dramatic change to switch the type. If this happens, the immune response may no longer protect fully, which means that your immune system might be able to recognize that antigen, might be able to respond to that antigen, but with a very low activity. That means it cannot respond fully and cannot protect fully due to the minor change. And this is not the worst thing. The even worse thing is the one we'll introduce later. That's the major change. This minor change can sometimes cause sporadic outbreaks or sometimes can cause limited epidemics. It does cause like major outbreak of flu, but can cause limited outbreak. Okay. Another one we'll introduce is the major change. This is the major antigenic changes in HA or NA resulting in the appearance of a new subtype, which means that this mutation is much bigger than the minor one. And this can make a new subtype of HA or NA. This occurs due to genetic exchange or reassortment. And this type of change can lead to the antibody, pre-existing antibodies, lose their function. The pre-existing antibodies do not protect anymore because the antigen changed. The antigen switched to a new type. So the pre-existing antibodies cannot work. So this major change sometimes can cause pandemic. So that's the global outbreak. For this one, we call it antigenic shift. So this shift means switch from one type to another. This is a major change. While the previous one, the minor change, we call the antigenic drift. Let me show you. This one. The minor change, we call it antigenic drift. Drift means there is a little bit change. That means change from one location to another. So that change is not big enough. So we call it drift, a little bit or minor change. While for the major change, it's like a switch or change from one type to another type, from one subtype to another subtype. This we call antigenic shift. Keep this in mind, the two definitions of antigenic shift and antigenic drift. This is the influenza pandemics ever happened in history or ever recorded in history. From 1918 to 1919, that's the one we call the Spanish flu. That happened 100 years ago. And for the estimated deaths worldwide, at that time there was no detailed record in every country, so we can only estimate the death. There was about 50 million deaths. So that's a lot. That's much worse than the coronavirus, which happened 100 years ago. Then after that, there are several other pandemics, including the one happened in the 1950s or 1960s. The most current one happened in 2009 to present. And this is due to swine flu, and it causes ongoing pandemic. I still remember the time in 2009 when I was in New York, and at that time this pandemic happens, and everyone was very nervous. And this pandemic also overlap with a flu season. So when you get the flu vaccine, you have to get two vaccines. One is this swine flu H1N1 vaccine. The other one is the seasonal flu vaccine. But the good news is that this pandemic is kind of controlled well, and it didn't cause a lot of death till now. And but we are not sure whether new flu virus is generating or not in the environment. We need to get prepared for another flu pandemic. And we need to get prepared, get protected every year by having vaccination. Maybe for like ordinary people, they don't have this sense to get a vaccine. But in the future, we are professionals, or you are professionals. Maybe you are a doctor, or you work in related fields in hospitals or in clinic. Try to tell everyone around you, try to tell your patients to get a flu vaccine if possible, if they can afford for it. That's good for both themselves, but also good for everyone in the world, so that we can set up herd immunity to protect everyone. Someone might be wondering, where do new HA or NA antigens come from? This is a big interesting story, because for the original of flu virus, scientists believe that originally flu virus is from birds, especially the birds in Southeast Asia. And at the very beginning, maybe they only infect birds and they don't infect human. But some time ago, somehow there is a spillover. So the virus began to infect human directly or indirectly. So this phenomenon we call spillover. For birds, there are some different types of HA or NA. The interesting thing is that for the pigs can be infected by the bird flu virus. The pigs can also be infected by human flu virus. So that means that in the pigs, it serves as a mixing vessel. It serves as a fermentation instrument. And there, the virus can reassort to form new types. And some of the new types can be pathogenic to human. So this is what happens. This is the birds. They can be infected by the bird flu influenza virus. And this is a human. They can be infected by the human virus. But for the pigs, it can mix them together in their body and then release new viral particles to infect human, which can be fatal, which can be deadly. So this is how the new NA or HA types come from. For treatment of influenza virus infection, we can design inhibitors, we can design medicines to target on the viral proteins, including the M2 ion channel inhibitor and also the NA inhibitor or other targets of the virus, so that you can design chemical medicine to inhibit those activity of those proteins. But for now, it's not big news. It's not a big good news is that those vaccines available, they only work at the early stage of the infection. So after exposure, those medicines can work within a short time window time, maybe 20 hours, 24 hours, or 48 hours before the replication of the virus in our body. Once they have replicated in our body, those vaccines does not work well, such as the NA inhibitor. So we need a better medicine to treat flu infection, and hopefully there might be some medicine coming out. Last year, I heard the news that in Japan, they developed a medicine which works better than the previous ones. And hopefully those medicine will be available to everyone. For prevention and control by vaccines, like we mentioned just now, vaccines are the most efficient way and low-cost way to prevent flu infection. But those vaccines are not always work well. It is difficult to prevent a flu by immunization because of the antigenic drift and antigenic shift in viruses. That's what we introduced before. Because after you make the vaccine, the viruses are changing all the time. The new viruses might not be the target of the vaccine. So we need to develop vaccines every year. We need to update the vaccines every year. So each year, scientists will choose a variant, a subtype of the virus, and predict that, use that for protection. Usually, they have a good guess, and those vaccines will work, but sometimes their estimation is not good, and they will off the target. So it is of importance if we can design a vaccine that can stimulate production of a broadly neutralizing antibody response effectively against many influenza subtypes. So that means the common vaccine or general vaccine. Some scientists are working on this area. In this area, they want to use some new strategy to design a new vaccine, and those vaccines can induce the production of antibody, and those antibody can recognize the common molecule, common site on different types of influenza viruses. Hopefully, we will have more vaccines available. Okay, so that's for influenza virus. Maybe we can have a break for like 10 minutes and we'll be back and move on to another topic. I do have more videos for you guys. We don't want to play it here, but you can find those videos and watch it on Superstar. Okay, we'll be